If you watch this video, you will know where origin of life research is at. And even if you are a non-scientist, you will be confident that you have the correct information on the matter. If you watched my previous 3 videos, you have a rich understanding of just how complex this thing called life is. Most of us don't. I certainly didn't until I took on this venture of making these videos. Here's the deal. If you follow science news as I do, you will have encountered hundreds of articles declaring that some aspect of origin of life has been solved, or may have been solved. If it weren't for the fact that the next article that comes along declares the same victory that the previous article declared, you may conclude that origin of life, abiogenesis, is settled science. Then you hear this voice crying in the wilderness declaring that origin of life scientists are clueless on origin of life. Clueless? Really? With all those articles declaring that clues have been found? And when there are two divergent declarations, how am I, as a non-scientist, supposed to determine which declaration is correct? Must I learn all there is to know about OOL research? Should I ask my high school biology teacher? Would he know? Let's listen to Dr. Robert Hazen, an origin of life researcher.
That if you decide you're going to go into this field, you better believe that this is true. And that's belief. This is not truth. It's just a philosophical stance.
Wow. He says you must believe, i.e., you need a faith position. Clearly, he doesn't think that this is a solved problem at all. Let's look into the literature. This was published in the journal Nature Review Chemistry. I know, 2017, ages ago. Not really. Remember, the Miller-Urey experiment, the serious beginning of OOL research, was in 1957. So this article shows where OOL is at after 60 years of research. Look way down in the bottom here, the current state of the field. Whoa. And it goes on to say that we need a major system innovation. That looks awfully like we're clueless. Now we see an area called chemical necessity. So this need for a major system innovation is well inside the pre-life territory. This is the area where Dr. Hazen has been working. It is possible that he has moved up the line a bit. As we follow the line up, we see fully alive. Okay, but we know that the JCVI SYN 3.0 is the simplest living organism known to man. So we know that there is no actual knowledge of where this line fully alive belongs. Could it be that the break lines belong below the fully alive point rather than above it? But are we really that clueless? Oh, heavens no, there are hypotheses all up and down this line. So we have lots of clues. What on earth, then, is Dr. Tour talking about? He says, look, nature needs to be able to produce these 5 phenomena. Useful polypeptide synthesis, useful nucleotide synthesis, useful polysaccharide synthesis, specified information, assembly of a cell. He placed this challenge out to the abiogenesis research community. His offer was most generous. If 3 top researchers agreed that any one of these challenges had been met, even in a painfully artificial environment where we are given all 19 canonical chiral amino acids, the nucleotides, the monosaccharides, all with 100% purity. This given is extremely generous. This is the kind of stuff that the Miller-Urey experiment makes, but by no means with 100% purity. Far, Far from it. Dr. Tour placed this challenge out to the OOL research community in August 24, 2023. His position: if a jury of 3 OOL researchers agreed that any of these challenges were met, he would remove all of his OOL is clueless content. As he has not removed this content, we can be assured that OOL scientists see all 5 of these to be unmet challenges. But here's the problem. Each of these are clues sitting on our chart. Most of these have published literature saying, we've got this bit, but obviously they don't. Each of these clues are potentially false clues. Do you remember our von Neumann life discovering a rock that was zapped with lightning which contained a strand of copper? Yes, he found a false clue. False clues are a real problem. If you think about it, some of the clues on the chart must be false clues. I mean, some researchers are saying that life began near hydrothermal vents. Others are saying that it started in the intertidal zone. Still others say it started between sheets of mica. At most, one of these hypotheses could be right. Therefore, at least 2 of these must be false clues. I remember back in the 80s when the RNA world hypothesis was all a buzz. This was so cool. Now the information was the duplicating machine itself. When we look at the 5 essential components of life, we have merged 2 of them, the computer and the information. I remember reading all manner of articles espousing wild things happening in RNA world labs and wondering why they kept calling it an hypothesis rather than a theory. It was an exciting time for OOL. But let me show you what Dr. Hazen has to say about the RNA world.
Well, the RNA world dilemma is that, it's a great way to potentially make copies of yourself. It's a great way to carry information. It's possibly a great way to catalyze reactions. But there's no known way to synthesize it. No one has been able to figure out a way how to do that. And so you really have to figure out, you've got the prebiotic soup with all these potential building blocks of life, and then you've got the RNA world, and that gap's a big one to fill.
He is motivated to believe in the RNA world because it doesn't challenge his research. So when he says that the RNA world hypothesis remains implausible after 30 years of experimentation with it, I've got to conclude that the RNA world is potentially yet another false clue. So here we sit. We've got clues all over the place. We know for sure that some of them are false clues. But we don't know that any of them are true clues. None of them. Well, we know that certain phenomena could happen in the prebiotic world, things that appear interesting. But we don't have any good scientific reason to believe that these phenomena were direct precursors to life. We only have faith. Because without faith, it is impossible to do OOL research. If we have no confirmed true clues, is Dr. Tour exaggerating when he declares that science is clueless on origin of life? I, for one, am with Dr. Tour on this one. So you say, but you promised to leave us non-scientists certain that we are getting the straight scoop from you. Ah, yes. How can you know that you've got the straight scoop? Watch what happens when we type origin of life or abiogenesis into Wikipedia. Look at the provided illustration. Remember, a picture is worth 1,000 words. What are these great big question marks doing in there? They are declaring that science is, well, clueless about origin of life. Let's try another tool designed to clarify science to non-scientists. Our friendly neighborhood AI bot. I asked Microsoft Copilot, has man made life in the lab? It responded, yes, scientists have made significant strides in creating synthetic life forms in the lab. Here are some key developments. 1. JCVI SYN3A. 2. Artificial DNA. Effectively, they added a man-made protein or two into the JCVI SYN. Please note that with JCVI SYN3A, they state, replaced it with DNA that was designed on a computer and synthesized in a lab. Well, you know that's an exaggeration, as Craig Venter's own website declares that 147 of the genes are necessary but have unknown function. They certainly can't have synthesized those in a lab. Clearly, while the DNA is a unique subset, the individual genes in the JCVI SYN 3.0 were all written by nature, not by man. So we can see that these sources are exaggerating. They are motivated to declare success. If they are saying that the issue of life's origin is not solved, we can be confident that it is not solved. So I asked Copilot, is origin of life understood by science? It responded, no. There are still many unknowns. How life began. The process by which life began is much less understood. There are several hypotheses. It then lists some hypotheses that are incompatible with each other. By point 5, it says, artificial life. Science have made significant strides in creating synthetic life forms in the lab, which can provide insight into how life might have originated. Again, they are referencing the JCVI SYN3 as the exemplar of where origin of life research is at. So please understand, these resources, Wikipedia and Copilot, are designed to communicate to the non-scientist. They are motivated to give you the most positive possible impression of where OOL is at. As long as they are saying that this is an unsolved problem, you can rest assured that it is. Clueless? As long as they are using the word hypothesis rather than theory, you can also rest assured that there is no confidence that the hypothesis is a true clue. As long as the only things available are questionable clues, I think it reasonable enough to declare that OOL scientists are clueless on origin of life. So am I suggesting that the only logical conclusion on this matter is that science is clueless on origin of life? I'm really not. However, if you understand that all known living organisms are just enormously complex, and if you understand that while science has many hypotheses about how it developed, it is with good reason that none are called theories, then your position is scientifically valid. If that doesn't translate for you as clueless, so be it. But please don't pretend that science has origin of life solved, or even nearly so. In my next videos, I will reveal all of object-oriented programming in DNA. I will show you examples where DNA implements procedures, if statements, while loops, classes and instances, encapsulation, polymorphism, inheritance, databases, and even abstraction, generalized algorithms. Some of these features, I am convinced, are way beyond what is possible within the Darwinian paradigm. I am convinced that what I am seeing proves that the Darwinian model is fundamentally inadequate as an explanation. The Canadian subarctic is beginning to thaw, which will draw my attention in other directions, so I'm not sure how quickly I will be able to get to this material, but it is very much my intent. Please like, subscribe, comment, and share this video. Thank you.